I'd like to welcome Professor Hillmen to this talk. Thank you very much for joining us, and I'll hand over to you, Peter.
Thank you, Brian. Thanks a lot for the invitation. It's great to speak to everybody. Hope you can hear me and see me clearly. What I thought I'd do is just try and touch some of the questions and topics. I'm going to start off talking about the new therapies, which there's a lot of interest in, obviously, and then move on to the trials, FLAIR and STATIC, and what trials mean. I think that and how they're designed, which probably is important to understand, and then talk a little bit about that, and then the announcement we heard about ibrutinib, which is very good news for all of our patients who may need therapy, and then the last few minutes talk about COVID-19, particularly the vaccinations, because it's a very rapidly moving area, which I'm sure many of you have a lot of questions about. So, and then we'll have plenty of time afterwards for questions if you wish. So I think the first thing to do, and I apologise, I know there's at least one or two patients that I've seen and probably have been through my drawings of CLL and understanding what's wrong in CLL. I think it's very important to understand what goes wrong with CLL cells, because that's where we've changed the treatment. That's why treatment is so much better now than it was a few years ago, and it's through a deep understanding of the disease. And that's important also for treatment choices as we go forward, I think, and will increasingly be so. So I'm going to, and also to understand, I guess, how people respond to vaccines, because CLL, of course, is effectively a disease of the immune system, and I think you need to understand a bit about the immune system. So first of all, this is sort of human biology, really. The immune system, the key cell in the immune system is the lymphocytes, and we're all familiar with lymphocytes, and they are very impressive cells, and that without an immune system that learns about the infections we've seen, we really don't survive. We wouldn't be here at all. So we need a very complex immune system to react to any bugs that we've seen or we will see throughout our lives, and to make sure we can fight them off. And that's very relevant to the current COVID situation. And so just to break that down a little bit, in the lymphocytes, there are really two main types of lymphocytes. There are B lymphocytes, or B cells, and there are T lymphocytes, or T cells, and you'll have heard some discussion around the COVID vaccines of those two types of cells. So in a normal blood without CLL, 90% of the lymphocytes are actually T cells, or actually slightly more, and only a small proportion are B cells. B cells make antibodies, so they will react to whatever infection we have or vaccine and produce a specific protein, a specific antibody that targets that, if you like, sticks onto that bug and tells the body to get rid of it. And for CLL, all or virtually all of our patients with CLL, the cell that's gone out of control is a B cell. So it's not the most common cell, it's a B cell that's gone out of control. But the T cells are at least as important as B cells in the immune response, because what the T cells do is several things. They first of all tell B cells whether they need to make an antibody, because we don't want to make antibodies against ourselves. So they sort of control the function of B cells. They also, in themselves, can target and eat bugs, so they can independently attack bugs and learn. You get T cells that are specific to COVID or measles or whatever bug we have. And so all of this action, if you like, between B cells and T cells happens in the lymph glands, the spleen, the bone marrow, and the liver to some extent. So they're the main immune systems. That's how it all sort of works together. So what happens if we now move on to CLL and understanding CLL? The problem with CLL is that one of those B cells is growing out of control. So it starts to duplicate itself, if you like, until it gets to many thousands of millions of cells. So an overgrowth of a single type of B cell. And we know from a lot of work done around the world, including in the UK, that those abnormal CLL cells that have become CLL cells are mimicking normal B cells. So they use the pathways that make a B cell work. So, for example, they have two major problems. One is that they are switched on to grow all the time. So normally a B cell will only grow when it's virus it's attacking or what bug it's attacking is around, and then will switch off when that bug's cured or got rid of. And so the in CLL, that pathway for proliferation, for growth, is continuously switched on. And then secondly, in the other important part of a B cell is once it's done all its work and killed the bug that you have, they commit suicide. So the cells kill themselves and get recycled. And that's, we call it apoptosis. And so what happens with CLL cells is they can't kill themselves properly. They continue to survive when they shouldn't. So you have this double whammy, if you like, that the cells are growing too quickly or constantly accumulating, and then they can't kill themselves. And so they're over, as you heard from John and many other patients, over time there's a slow accumulation of these cells which push out the normal cells. So that's how they're the main issues in CLL. So understanding that process, understanding why CLL cells are growing too much, why they don't die, has allowed us to develop therapies that specifically switch off those pathways. So I tend to liken it a bit like so chemotherapy, and unfortunately with the announcement today, chemotherapy for probably every patient, and we're going to talk about it with CLL, is no longer required because we have targeted treatment. So chemotherapy is like knocking if you think of a padlock, is like attacking it with a hammer to try and knock it into remission, which works for some of our treatments. Whereas the new treatments switch off the pathways. It's like having a key that switch off the pathway specifically, either the proliferation pathway or the cell death pathway. So that's allowed us to develop these new therapies. The first one in the if you like, the cell growth side was ibrutinib, and now acalabrutinib, as we heard, which are specific inhibitors of that pathway. And then venetoclax, which encourages the cells specifically to kill themselves, so to not accumulate. And so they're the two main pathways that we can target. And that's made a massive difference to our patients. And you heard John's story, but there's many other stories like that where patients have really benefited from this sort of targeted approach for treatment. So if we think about developing these therapies, I mean, I already explained the complexity of trying to identify the target, if you like. How do we get these drugs to be approved and to be used for patients and prove that they work? Well, we do that through clinical trials, and many of you will have been exposed to clinical trials. I've obviously been running and designing trials since my first CLL patient in 1996. So we've done a lot of trials over that time. And you can think about trials there's several different phases of clinical trials. So the first phase is this potential drug, say ibrutinib in 2009, it would have been, is this drug safe to give to people, and does it have any sign of effectiveness, and what dose should we use? So the first early trials will look at the right dose and do have any evidence that our theories that this drug would work. The second, so that's the first phase 1, phase 2, we would call them. After we have that, a company these are usually made by pharmaceutical companies, will do what's called a phase 3 trial. So that means that we want to show that a drug works. And so we'll take the standard treatment, in this case maybe FCR or chemotherapy, parameter-based treatment, and we'll compare that to, in this case, ibrutinib or whatever new drug there is, in a randomized way. So that will typically involve hundreds of patients around the world for these commercial trials. They're designed really to show that this therapy is better than the current treatments, and that will lead to the approval of the drug by, in America, the FDA, and in the UK, the MHRA or the EMA in Europe. And we've heard these, because these are the same bodies that are approving vaccines, so we've heard about them more recently. And so that process usually takes several years before we get an approval. But by the time we get approval from the phase 3 trial, we've only got relatively limited experience of the drug. So we might have 3 or 4 years experience. And so of course, some drugs like ibrutinib may be used for a much longer period than that, but we don't have any safety data or efficacy data beyond that when we first get approval. So, and they're relatively small trials. So the next important trial, which FLAIR comes into the bucket of, is our what we'd call academic or collaborative group trials. So that would be a trial that we would run, for example, in the UK through the NCRI group, which is basically all the hospitals in the country, and we would design a trial which would probably treat more patients and ask more important, well, not they're all important questions, but more detailed questions about which patients benefit the most. So whereas the randomized trials in frontline for ibrutinib, for example, had 200 to 300 patients, FLAIR, which is will have about 1,500 patients now included, so it gives us a lot more information. Just to show you the challenges we have in designing the trials, of course, ibrutinib was first used in 2009 in the first ever trials. We started discussing FLAIR with the companies that are making the drug in 2011, and I don't think it was approved, the drug was approved till 2014. So we were planning FLAIR from 2011. We got approval through all of the hurdles towards the end of 2012, November 2012, I looked it up today, and we didn't open the trial because there's a challenge to get protocols written, sites open, lots of other agreements that have to be had until towards the end of 2014. So we designed a trial then. FLAIR, just to use as an example, we started off with just comparing ibrutinib rituximab with FCR chemotherapy in 700 plus patients. That finished some time ago, but we were able to adapt it to add in even new combinations like the combination of ibrutinib and venetoclax. I may talk about that a little bit later on, but it gives us a lot more scope. And what the trial does is gave access to many patients in the UK, potentially much better drugs and proven to be better drugs, I think, much earlier than we would have in the real world with approvals and all the funding issues that we deal with. So in frontline, ibrutinib wasn't approved for several years after the 2014. So that's the way the trials work. The trial, FLAIR finished recruitment into its main part of the trial, I think this month or last month, with just under 1,500 patients. We still have some patients with 17p porous disease going into the trial, and we can talk about that later if we wish. So you can imagine the challenges of designing a trial like that. So how long do we use ibrutinib for? It was one of the key questions. When we only, at the time when we designed the trial, had 2 or 3 years experience in only relapsed patients. So nobody really knew how long we should use ibrutinib for. When the drug was approved, we only had about 3 years experience of ibrutinib in the phase 3 trials, but it was approved for continuous use until disease progression, which could be decades in patients potentially if people respond well. So the approval really looks at the early responses, but we actually get approval which goes on for much longer. And so in FLAIR, when we had a lot of discussion, obviously, we designed a trial to have 6 years of treatment on the basis that we would either consider what we would do after FLAIR when we got nearer the time with more information, because we'd have 7 or 8 years information, or that it would be appropriate to stop treatment then and see how patients got on, because we didn't know. And so we've been having a lot of discussions about what do we do beyond the 6-year period in FLAIR. So what's emerged, I think, in terms of ibrutinib over that time is, first of all, it's a very effective drug, as is venetoclax. I mean, we see really, from my experience treating CLL over many, many decades now, I guess, many years, phenomenal responses and outcomes for many patients with CLL, particularly when it's used early on in the disease in frontline. So, I mean, the first thing to say is it's a very effective and a very safe drug. So, first of all. Secondly, we do see the emergence of resistance to the drug. So patients who have been on the drug for a long time, the disease can escape from control. Why does that happen? Well, first of all, it tends to happen more in patients who've had heavy pre-treatment than the people who've had no treatment before. So up to a third of patients, or maybe a bit higher, who've had chemotherapy treatments before may become resistant over years on ibrutinib. Less so early on with frontline, but we don't know later on. One of the main reasons people could become resistant is because the key, if you like, where the ibrutinib fits in and switches off the pathway, if you like, the lock changes itself, so the key no longer fits, and so the drug now no longer can block a mutant part of the CLL, which then is a selective for it, like the variant COVIDs, I suppose, that they grow out of control and then are resistant to that type of treatment. And that only really happens when you're on treatment. So there's a strong rationale for thinking about treatment breaks to stop that resistance occurring, so that when a patient eventually progresses, if it may be years later, they would still be responsive to the same treatment, most likely. The second thing which is emerging, and ibrutinib may be an important way to address this, is the side effects we see with ibrutinib over a long time. So a side effect from a drug, if you're only taking it for 6 months or 12 months, is sort of tolerable most of the time, as long as it's not too severe. But if you're taking it indefinitely, even the nuisance side effects we sometimes see are a problem. So my take on ibrutinib is that 80% of patients have very few side effects at all. So they tolerate the drug very well, and they respond very well to treatment. But probably about 20%, maybe a bit more as we go later on, have some forms of side effects. And that's because, in part at least, the drug, although it switches off the CLL cells, also isn't entirely specific, so it switches off a few other things that are important, that can cause some side effects. And the ones that we probably worry about the most from a doctor's perspective is high blood pressure and irregular heartbeats, some of which are very serious. So that happens certainly the irregular heartbeats happens in maybe 10%, maybe a bit higher percentage of patients, particularly in patients who've had previous problems with their heart. And high blood pressure, depending on the series, happens in a bigger proportion of patients. So it just shows there's something we have to be at least aware of, especially when patients are on treatment for a prolonged period of time. So they're the main issues in terms of having indefinite therapy, and particularly on the basis that we don't know we haven't got data beyond 10 years of ibrutinib now, and patients will stay on for longer. Just a word about the development of resistance. We have new drugs coming through that will target that resistance, and we have not just venetoclax but other drugs similar to ibrutinib that appear to have promise in patients who have become resistant. So we aren't stopping the development of drugs; they continue. And secondly, in terms of the safety profile for those patients who have a lot of nuisance side effects that affect their quality of life, like arthralgia and other issues, the new generation or second generation BTK inhibitors, which are more specific, like acalabrutinib, may help, although we still have more trials coming through this year and beyond to see exactly how much benefit this more specific inhibitors have over ibrutinib. But I have to remind you that they're all very effective. And then finally, just from the FLAIR trial, the combination of venetoclax and ibrutinib, there's lots of work looking at those combinations to see if we can actually eradicate the disease and get a very durable remission, even an indefinite remission. We've done trials ourselves, and they've been done elsewhere, and the FLAIR trial is a big trial which has included that combination in the second part of FLAIR. So those results will come out a little bit later on, but are probably an important next step towards the treatment and hopefully moving towards a potentially curative approach to CLL. So we have this issue that we don't know how long to use drugs like ibrutinib, and there's been a lot of work in cancer generally looking at these types of drugs to see, should we use drugs indefinitely, or should we have treatment holidays and then re-expose patients to treatment? And actually, there's quite a lot of work in other cancers that intermittent treatment of that type can be an effective way of avoiding resistance, of avoiding toxicity or controlling toxicity, and potentially, obviously, from a cost point of view, it would be cost effective. And so we've been discussing this a lot, and that's where STATIC came from. So the STATIC is one of the next trials we'll do in the UK. It's now got full approval eventually. And what STATIC will do will include patients from FLAIR who have been on ibrutinib, and also some patients who are on ibrutinib who aren't in FLAIR. So the patients who have had ibrutinib in the relapse setting, so they've had chemotherapy perhaps before and now treated with ibrutinib and are now in remission. So patients will have to have been on ibrutinib for at least well, 6 years in FLAIR, or at least 2 years if they're in the relapsed refractory group of patients, and they have to be in a good remission, so they have to be in a deep remission, and that means basically the blood count and no disease that we can feel or the patient reports, at least for a year of treatment in remission for that long. And then what we plan to do is we will compare continuous treatment with ibrutinib, so continuing on the drug that patients are on, or pausing therapy, and then as soon as we see any sign of the CLL coming back, to retreat the patient with ibrutinib again, so to restart the treatment and potentially stop and restart to use intermittently. And that has a potential benefit of meaning people will remain sensitive to the treatment, so we hope it will reduce, and we expect it might reduce resistance, which obviously will improve the outcome for patients, also reduce the side effect, the toxicity, if you like, that any patients might be experiencing, whether it be very mild or more serious. And potentially obviously have cost effectiveness advantages as well, but that's a very secondary importance. And so the advantage of static is that if people are randomized to the intermittent treatment, there'll be a randomized trial, there'll be 800 patients in total, 400 in each arm, then we know that we can and we will restart treatment as soon as we see any evidence of the CLL coming back. And the advantage of that is we would expect, because we won't be waiting for them to progress like we would in first treatment or subsequent treatments, we would expect the disease to come under control again, because it's not seen ibrutinib for hopefully quite a period of time. We know from several studies actually now that if you stop ibrutinib in patients in a good remission, and most of this has been done because people have side effects, that there's usually a prolonged period of time before the disease becomes active again. So it can be an average of 2 or more years before we see any sign of the disease coming back. So we are hopeful that patients will have a prolonged period of time off treatment before restarting. So static should be opening hopefully in the summer. We're trying to do it as quickly as possible. We've been negotiating it for a long time. There will be some patients in FLAIR who finish ibrutinib before static opens in FLAIR, but they will also be eligible, and patients would be able to choose to stay on ibrutinib if they want within static, we hope, but it's logical to go on to the randomized intermittent treatment. So we can discuss static a little bit later on. And now moving on, finally, in terms of the treatment, acalabrutinib. So the announcement which we had, Brian showed in his introduction for acalabrutinib is really an important development, because at the moment in the treatment of CLL, in frontline previously untreated patients who need treatment, we wouldn't treat anyone any earlier. We had until now for the most patients just venetoclax and obinutuzumab available for all patients for a year of treatment, or chemotherapy for the younger patients, FCR-based treatments as the option. And acalabrutinib or ibrutinib have been major advances in the treatment of CLL, and now acalabrutinib is now available in frontline for patients who would not be eligible for FCR. That's where the trial was done, and that's where the approval was gained, and we've treated a lot of patients in those trials. So we now have non-chemotherapy options for all patients with CLL. For the younger patients, venetoclax plus obinutuzumab, those who would be fit for FCR, and the ones who wouldn't be fit for FCR, that combination, or acalabrutinib. And for the 17p, which is a small proportion, we have either ibrutinib or acalabrutinib available for those patients. So that's a really important change, I think, in our available therapies, and allows us to choose patients to choose which is the most appropriate treatment for them with the help of their clinicians. So finally, in the last 5 minutes, I'll just talk about COVID vaccinations, because that's been most of my consultations in the last, certainly a year have been about COVID, in the last 2 or 3 months have been about vaccinations. So it's obviously a very hot topic at the moment, and particularly as there was a paper, the first paper we have in terms of response to COVID vaccines in cancer patients generally, but some hematology patients came out last week and hit the news. So first of all, it's gratifying to see that most patients from the survey have first of all been offered the vaccine and had the vaccine. I was delighted to see that the very few patients didn't want to have the vaccine. I think that's a really important sort of message to get across, because the vaccines are certainly safe in CLL patients. We've treated many patients have had vaccinations with AstraZeneca and the BioNTech, and we can talk about, I'm sure, the side effects a bit later on with the thrombosis story recently breaking around Europe. The issue for us, I think, for our patients is are patients going to respond as well as somebody without CLL to the vaccines, and what does it mean in terms of protecting our patients, and the fact that shielding is coming to an end, and can people behave as normal? So first of all, you would expect that even in CLL without treatment, that because it's a disease of the immune system, that they may have a suppression of their immunity, and therefore the response to vaccines may not be as good and may need to be studied more closely. And certainly patients who have had or on treatment, the treatment, because it's designed to kill lymphocytes or to suppress the activity of lymphocytes in some of the newer therapies, inevitably we would have a concern that this might affect responses to vaccines. So we've been concerned about this for some time. As I said, the vaccines are not dangerous in terms of not live vaccines, so I'm strongly of the opinion that patients should have the vaccine when offered. So the paper that came out, I'll just first of all talk about blood cancer. The Blood Cancer UK, which is used to be Bloodwise, has set up a vaccine task force, which we meet every Wednesday evening to talk about the studies ongoing in all haematology patients. There are 13 studies in the UK looking at response to vaccination. If we're thinking about vaccine response, the vaccine mimics what happens in a normal immune system. And as I said to you before, the easy measurable bit is the antibodies to the vaccine. So the anti-COVID antibodies, or serology, are the antibodies that you make in response to either infection or a vaccine. But equally, possibly even more important, is the T cell response. So are the T cells responding to the bug or the vaccine? And then finally, can you measure whether the response is neutralizing any virus infection? So there's three main ways you can look at a vaccine response, and the one that is available, the serology test, only tells us a little bit of the response for patients. And so with one of the advantages of the Blood Cancer UK vaccine task force is we're standardizing the assays across all our patient groups, including CLL, so patients with CLL. So using those three techniques, T cells and serology. There's a large study which is being run from Paul Moss in Birmingham with 600, I think, CLL patients, including some from FLAIR. And we're trying to get permission to collect information from our FLAIR patients in terms of response to vaccines. The study that came out of King's and Guy's Thomas's from the Crick last week was in cancer patients. I think there were 151 cancer patients that they looked at, and looked at the response to mainly one vaccine, because they were given in routine sort of use here, and then some with the second vaccine. And of those 151 patients, I think about 51 patients had hematological leukemias or lymphomas or blood cancers, and I think only about 11 of those patients, I'm told, had CLL. So we have little data in terms of CLL specifically, and we don't know if they were untreated or treated. The response in that trial for healthy control, so they had a cohort of healthy control people, was over 95% of people had an antibody and a T cell response to the vaccine. And across the board, and for cancer patients with a single shot only the BioNTech was studied in this particular study. There's no data on the AstraZeneca. About 39% of solid cancer patients responded with an antibody level, but more responded with T cells when they did the T cell analysis. And only 13% of the hematology patients responded with antibodies to the first shot of the BioNTech vaccine. More again with T cells, so about half the patients had some evidence of T cell response in the hematology side. So that wasn't broken down by CLL or not CLL particularly. The second vaccine seems to increase the response. So in solid cancers and in the hematologic cancers, there's a higher response rate when you look at both the serology and T cells after the second one. So we've been lobbying the JCVI committee to see if we can get our patients with not as long a gap between the vaccines, and so far they haven't agreed for us to do that. Although last night we were talking about it again, and there are there's lots of pressure being applied to try and allow us to do that. In a non-CLL patient, it's likely that the 12-week gap is fine, and that you see good responses at 12 weeks when it's given again, and we know that from the real-world data, but we obviously are more worried about our patients. In terms of the two vaccines, there's no evidence that I'm aware of that either the AstraZeneca or the BioNTech is any better in terms of effectiveness. In terms of the side effects, and obviously thrombosis has been all over the news recently. The rate of thrombosis is pretty low considering the millions of vaccines that are being given. We see thrombosis anyway in the population. This is the sort of thing we have to collect in trials and beyond, and we have to we can't just assume because it's common in the population that the vaccine isn't causing a problem. But so we have to collect the data, so that's really important. We have to be vigilant. But given the risk of COVID and given the risk that we know of the vaccine, there's no doubt that in my mind that the benefits of having the vaccine far outweigh the risks that we're seeing with AstraZeneca, and I think it's damaging to the response to pause AstraZeneca use is damaging to vaccines generally. So I think I've covered most of the things I said. Probably went on a little bit too long, Brian, but I'm happy to take any questions.